This is a production of Cornell University.
All right, well, thank you so much, Jeff, for the introduction. I'm really excited to be here with y'all today. Like Jeff said, I'm Elizabeth McCarthy. My pronouns are she, her, hers. And yeah, my love affair with Nicotiana, Valentine's Day, we're going to talk about love of plants, started when I was an undergrad, and I never want to leave it. I took a little bit of a hiatus occasionally, but not very much. I'm excited to talk to you about how polyploidy and hybridization affect flower evolution in these awesome species. There. All right, so I'm going to start with an introduction, and then I'm going to tell you two stories about flower color evolution in polyploids. One on a young polyploid that's involving homeolog bias and subgenome dominance, and one on some older polyploids that involves some transgressive delphinidin production. And then I'm going to finish up with some of the things that we're doing in the lab right now so you can see what my awesome undergrads at SUNY-Cortland are doing. So allopolyploidy involves two things, both whole genome duplication and interspecific hybridization. So as we can see up here, we have two different diploid species. Here are their sets of chromosomes, different colors, and they come together to create hybrids that have 4 sets of chromosomes, but 2 different diverged genomes back in the same nucleus. And this leads to this genomic shock, where they have to figure out how to work with each other. And this leads to all sorts of different changes, but can also lead to changes in phenotype, like we can see in these polyploids here. And so I'm interested in how these phenomena lead to differences in flower phenotypes. Polyploidy is everywhere, especially in land plants. So we have ancient polyploidy, at least in all seed plants, another in all flowering plants. And when we have so many genomes access that we have now, we can see that all of these red lineages are whole genome duplications across land plants. So we have ancient polyploidy everywhere. On the flower side of things, we know that morphology and color is going to affect the type of pollinators that interact with these flowers. So here are just a subset of pollinators, and you can see that the flowers that they're visiting have different colors, different shapes. They open at different times. If we could smell them, they would smell different. So we have this really exciting system where we can look at differences, not only in the plants, but see how they're interacting with animal pollinators. And we're going to see this pathway a lot. Don't worry, it's coming back. And this is the flavonoid biosynthetic pathway, which is a branch pathway with many enzymes working together. So the enzymes are in bold by the arrows, and they produce all of these different types of pigments that lead to flower colors. So on the bottom down here, we have our anthocyanins. Those are colorful. We have red pelargonidin, pink or magenta cyanidin, and purple delphinidin. And then up here, we have flavanols. These are colorless to humans, but they absorb UV. So differences in flavanols can give differences in signals to pollinators that have UV receptors. So all of them are important for the formations of flower colors and how they interact with pollinators. And we also have some understanding of some transcription factors that are promoting the expression of some of these flavonoid biosynthetic pathway genes. So my overarching research questions in my research program are, again, how does this genome merger that we get with hybridization yield the novel and diverse phenotypes that we observe in allopolyploids? And then how do those phenotypes affect the evolution of allopolyploid species? And Nicotiana is a fantastic system to study this in, because we not only have an amazing diversity of flowers, size, shape, color, but we also have a ton of polyploids, and they're all different ages. So we have some that are young that have not undergone speciation, that are all less than 1 million years old. We have another section that's about 1 or 1.5 million years old that has speciated into a few species. Another that is 4 million years old that is speciated into a few more. I ran out of space on this slide. There's yet another one that is 6 million years old that we think has maybe speciated into potentially 90 different species. What? So there's a whole lot of stuff going on here. We have this really nice trajectory of different aged polyploids, so we can ask questions about the differences between what's happening short-term and long-term consequences of allopolyploids as they evolve. Today, the stories I'm going to tell you are involving Nicotiana tabacum, this young polyploid, and Section repande, this older one. So we're going to get a flavor of a couple of these things. All right, so that first story I'm going to tell you about is about Nicotiana tabacum and homeolog bias and subgenome dominance and how it affects flower color. So Nicotiana tabacum is the product of Nicotiana sylvestris, maternal progenitor, and Nicotiana tomentosiformis, paternal progenitor. We have these natural polyploids, like I showed you already, that are 0.6 million years old. And we have some different accessions that have some variation in flower color. We have these really bright magenta ones and some light pink ones. But we also have synthetic polyploids that we have created, not me, I use them, other folks, have created in the lab, where we have first generation polyploids, so we can get an idea of what's happening immediately following polyploidy, to then compare that to what's happening over evolution in our natural ones. And we, again, even in our synthetic lines, see some variation in flower color. So we have light pink ones and we have darker pink ones. So we have a great system that we can compare. Again, flavonoid biosynthetic pathway. The differences we see in these pinks is mainly due to differences in cyanidin pigments, so that's the focus we're going to think about for this story at least. And we're also going to think about homolog expression bias. So when we have allopolyploids, we have homologs, two different progenitor copies inside our genome. And so, for example, DFR, which is one of our genes in our pathway, will have a maternal copy and a paternal copy here in purple and teal. And we can have either equal homolog expression, where there's equal expression from both of these copies, or we could have unequal expression, and then we call that homolog expression bias. So for example, down here, we have more copies or more transcript of our paternal copy than our maternal copy. So that would be an expression bias towards the paternal over the maternal. And in the talk, I'm going to be reporting that homolog expression bias as the percent of our maternal homolog that is present, so the proportion of sylvestris in these polyploids. That's at the gene level. So we're thinking about how are the, what's the ratio of our two progenitor copies? If we build that up to the whole transcriptome level, we can see whether we have a skew in the bias. Do we have more sylvestris bias genes than tomentosiformis bias genes, or do we have an equal number of each? So if we do have a bias, we call that subgenome dominance, where one progenitor genome has more expression overall than the other. Okay, so my questions for this story are, what's the genetic basis of the pigment and flower color differences that we observe in these tabacum allopolyploids? And do those correlate with homolog bias differences in the polyploids? So do we see that these flower color differences are associated with homolog bias differences? So to answer this question, this is part of my postdoc work with Amy Litt at UC Riverside. We did a developmental series for all of our different accessions. So we have, and we collected corolla tissue from early buds, mid buds, and late floral buds across all of our different, our progenitors, our natural polyploids, and our synthetic polyploids. And this work was done by two undergrads, Amber Lahorn and Imelda Curti at UC Riverside with me. We did a transcriptome analysis from these accessions with an Illumina platform and a 1x85 reads. We used a de novo assembly approach to analyze that data, but we found that these short reads made it really difficult to distinguish between our two progenitor copies. And so when I got here, y'all might recognize him, Jacob Landis. You'll see his face a couple times. So in collaboration with Jacob, we added some long-read Oxford nanopore sequencing to our progenitors to be able to increase the quality of our assemblies. We made those diploid assemblies, put them together in silico to create our allopolyploid assembly. And with that long-read data, we were able to distinguish between our two copies. So doing great. So we are looking at our different biosynthetic pathway genes. I told you you were going to see this pathway a lot. I've color-coded it so we can have an idea of where the different genes are. So the colors in the pathway are related to the colors in our points. The points on these graphs are our flavonoid biosynthetic pathway genes. The violin plots behind them are the transcriptome overall. So in this graph, we're looking at differentially expressed genes. So on our x-axis, we have our different pairwise comparisons. So between our progenitors, between our polyploids and our two different progenitors, and among our different polyploids. And then on our y-axis, we have our log two-fold change of our differentially expressed genes. Another thing to notice is that our bold colored points are our tomentosiformis homologs, and our transparent points are our sylvestris homologs. So if we look, this is in our early development, so our early bud, seeing what's going on there. If we look at our comparison between our progenitors, we see that most of our genes are differentially expressed, just what we want to see. So our sylvestris homologs are upregulated in sylvestris, our tomentosiformis homologs are upregulated in tomentosiformis. When we compare our tobaccums, our polyploids, to our maternal progenitor, unsurprisingly, we see that our paternal homologs are upregulated in the polyploids compared to their maternal progenitor. But we also see that we have some maternal homologs that are upregulated in the polyploids compared to their progenitor. Similarly, when we compare to our paternal progenitor, we again see that the maternal copies are upregulated in our polyploids. But in this case, when we think about the paternal homologs, they tend to be more highly expressed in the progenitor compared to the polyploid. And then when we compare among our different types of polyploids, we are starting to really tease out differences that might be involved in the differences in flower color we see. When we look at comparisons between light pink and magenta flowers, we're starting to see genes that are involved in producing anthocyanins. The black points here are our FLS gene, which gives us towards flavanols, and we see differences among our accessions here. So we're starting to get some of these pieces that might be important in creating these differences in flower phenotype. And so we looked at that a little further. We took these different comparisons where we had light pink and dark pink polyploids compared to each other, and we saw whether there were any commonly differentially expressed genes to those comparisons. And so we found a few. At least 2 comparisons had an upregulation of DFR, which is again this one that is the first gene towards making anthocyanins. It was upregulated in the dark pink phenotype, whereas FLS, which is towards making flavanols, is upregulated in our light pink things. And then we had a handful of other genes that were singletons, but again, they seem to be concentrated in the genes that are producing anthocyanins, and they're upregulated in our dark pink things, or flavanols are at the beginning of our pathway for our light pink phenotypes. So then we took a step back and we're asking this question about, do changes in homolog bias in these genes correlate with the differences in flower color that we observe? And so for these plots, on our x-axis, we have our developmental time points. So these are our early, mid, and late tissues. And then we're looking at our percent sylvestris. So that's our homolog bias. So if it's 0.5, that's equal. If it's above 0.5, that's sylvestris biased. If it's below 0.5, that's tomentosiformis biased. And our sylvestris and tomentosiformis are behaving excellent. We like to see that. But in this case, in FLS, we see that we have pretty equal expression between our 2 progenitors, and that seems to be the case for all of the accessions that we looked at. We did see some differences in homolog expression bias, but they did not correlate with phenotype, like we hypothesized. They instead correlated with the polyploid age. So our synthetic polyploids, which are here in these pink and purple points, tended to be more equally expressed, whereas our natural polyploids here in cyan and orange are further away from that equal expression. So those are more biased. And we saw that both in CHI at the beginning of the pathway and in DFR towards making anthocyanins. And we found this overall. We found that natural polyploids tended to display stronger homolog bias, so more different than equal, both in our flavonoid biosynthetic pathway genes that we can see here. So we have an increased distance from equal in our natural polyploids compared to our synthetics, and that also held across the entire transcriptome. So we're seeing we have stronger bias in our natural polyploids compared to our synthetic polyploids. So then we can ask this question. We looked at individual genes. What about overall? Do we see subgenome dominance where we have more sylvestris bias or more tomentosiformis bias genes than the other? And in this plot, we have our progenitor ratio on the x-axis and our polyploid homolog ratio on our y-axis. Then we have a lot of points in here. So I also put these histogram curves along the sides to give us an idea of the distribution of those points. And we can see that our progenitor ratios and most of the ratios of our polyploids are pretty balanced. We have a relatively symmetrical curve here. But we see this pink one, we have this line that is a little bit higher on one side than the other. So QM24 seems to have a little bit of a bias towards sylvestrous biased homologs over tomentosiformis. And if we look at their floral phenotypes, here's QM24, here are the two parents. You can see that QM24 shares some things with sylvestrous that may be a little bit further away from tomentosiformis. It has a paler color, it has a smaller tube opening, it's more star-shaped, and so maybe that is reflected in the morphology that we observe. Okay, so to really answer that question of whether we have subgenome dominance, what I did is I partitioned or grouped these genes into either a sylvestrous bias group or a tomentosiformis bias group, and then did a chi-square test to see whether we had a skew in where those genes fell. But we did this in several different thresholds. So first, naively, I think, I just split it down the middle. And if it was bigger than 50%, I put it in sylvestrous bias. If it was smaller than 50%, I put it in tomentosiformis bias. But that gives us no equal expression. And so we created this buffer zone where we have equal expression. So we have a threshold where it was considered sylvestrous bias if it was 55% or above, and tomentosiformis bias if it was 45% or below, and then also another threshold where it was 60% versus less than 40%. Everybody feeling good about that? Can you tell that I'm an undergrad person? Yes, awesome. And then we took those the numbers that are falling in those categories to do the chi-square test to see if we actually had subgenome dominance. And what we found, so here on our x-axis, we have our progenitors, we have our different polyploids, and then on our y-axis, we have bias. So our dotted line in the middle is no bias, and above the line is a tomentosiformis bias, and below the line is a sylvestrous bias. So what we see is that we have a slight bias across the genome toward tomentosiformis in our progenitor ratio, a slight bias towards sylvestrous in QM24, like we saw in our other plot, but no bias in QM25 or our natural polyploids. So a little bit of subgenome dominance here, but not a whole lot. Okay, but that's the whole genome. Let's look at our flavonol biosynthetic pathway genes. So we've seen this type of graph before. We have our progenitor ratio on the x-axis, our polyploid homolog ratio on our y-axis. The gray points are across the genome, or transcriptome, excuse me, like we saw before. But now we're highlighting where our flavonol biosynthetic pathway genes are falling out. Again, they're color-coded based on our pathway. And what we can see is that especially in our progenitor ratio, we have a huge bias towards tomentosiformis. We also have one in our polyploids, but it seems to be a little bit less. So let's look at that in a slightly easier way. We can see that our bias has shifted a lot here, right? So we have a huge bias towards tomentosiformis in our progenitors, in our flavonoid biosynthetic pathway genes. But that is still present, but greatly reduced in our polyploids. So we still see this tomentosiformis bias, but it is way less present. Mostly our synthetic polyploids are unbiased, but we do see that sylvestrous, the whole genome of sylvestrous is playing a role in determining the flower color of these polyploids since we have that reduction in the bias. So even though sylvestrous does not make pink colors, it is involved in making pink, its genome is involved in making pink colors in polyploids. So both of these genomes are working together to create our differences that we see in polyploids. Okay. Again with these plots, so a new thing that we can think about. Where things fall in these plots gives us an idea of their regulation. So if things fall out in this horizontal line, then they are likely transregulated because they're in the same environment, and so they're going to have equal expression. Our diagonal line represents our cis regulatory differences because those cis-regulatory differences should track with what we see in the progenitors to what we see in the polyploids, giving us that diagonal line. And then anywhere else in the plot is likely some sort of cis-trans interaction. And what we can see from this, again, we have our flavonoid biosynthetic pathway genes pulled out here. Here they're color-coded by gene. Here they're color-coded by our accession. So again, our pink and our purple are our synthetic ones, and our cyan and our orange are the natural ones. And what we see is that often when we see differences, the synthetic polyploids seem to have more equal expression, and then our natural polyploids have more deviation. And so this seems to suggest that our natural polyploids are evolving cis-regulatory differences and cis-regulatory changes as they're aging, based on what we're seeing in the synthetics, which I thought was pretty cool. When we were looking at the sequences of these genes, we also found that a few of our tobacco accessions had a frameshift mutation in DFR, which again is that first gene towards making anthocyanins. And we have a 1 base pair deletion leading to this frameshift. Other folks found this also and did some functional work on it, and they found that that frameshift led to a non-functional gene. So when we have the frameshift and we bombard those constructs into a white tobacco mutant, the gene that doesn't have a frameshift is able to create anthocyanins. You can see that in this cell. There's our control because our YFP is there. We know that our constructs are in there. But when we have a frameshift, again, we know this cell has that construct, but it can't produce any anthocyanins. And so we know that this frameshift is non-functional. So we seem to see that this DFR homolog in the Sylvestris copy might be becoming a pseudogene, even in young polyploids. And also, because of this, we know it's non-functional. We did not include that transcript in our overall transcript analyses, which I'm going to show you next. Because we found that our homolog bias did not correlate with flower color, but when we pull our expression together, we do see that overall expression of these genes does correlate with flower color. So you've heard about FLS, which is making flavanols, and DFR, which is making anthocyanins. We're looking at those expression of those genes across development in these plots. But we can also look at the ratio here because they're competing for the same substrates in the pathway. And so what we noticed when we looked at that ratio is that we have an elevated FLS to DFR ratio early on in development in our 2 polyploids that have a light pink phenotype, which you can see here. And so we think that that is driving the flux of the pathway towards making flavanols at the expense of making anthocyanins. And it also seems to be because DFR isn't expressed as much early in development. It's being turned on later, and it's giving us that elevated FLS to DFR ratio. But because we have this competition, having more FLS means that we're going to have more of our pathway going towards making flavanols, less going towards anthocyanins, giving us that light pink phenotype. We also looked at the sequence variation in this gene. So this is work that has been done by Abigail McCoy, who just graduated last semester. She worked with me for 7 semesters at Cortland, and she was doing another project that I'll tell you a little bit about at the end. But what we could see from that is that we do have some variation, especially in regions that are important in which substrates this gene is interacting with, because it works on several different ones. And so sequence variation may also affect how these enzymes are functioning. And so it seems like there's both a transcription and expression story to this story, and potentially a sequence evolution story to this story. Okay, so conclude on this story, we found that homolog bias does not correlate with the differences in flower color that we observe. We do see that we have a stronger homolog bias in our natural polyploids compared to our synthetic ones. We have a slight subgenome dominance in TUM24, but not the others. We have a reduction of tomentosiformis bias in our flavonoid biosynthetic pathway genes and polyploids compared to their progenitors, and that transregulatory differences are important early on in polyploid evolution, but perhaps our natural polyploids are evolving more dysregulatory differences as they age. We've got this potential pseudogene popping up, and that our high FLS to DFR ratio is potentially driving our late pig flower phenotype, and that our pigments that we see that are different across these species are probably due both to transcript levels and also the efficiency of these enzymes. All right. So let's shift gears. We're going to talk about transgressive delphinidin in these older polyploids. So here's Nicotiana, section repanda. You've seen Sylvestris before. She gets around. And then obtusifolia is our paternal parent here. And this is a single event that happened 4 million years ago that has speciated into a few different polyploids. And we also see, again, flavonoids. We're going to talk about our pathway. This one is going to be a flavanol, so camphorol and quercetin, and a delphinidin story. Because you can see these purple arrowheads here. Nicotiana and repanda have delphinidin, the purple pigment, but Sylvestris and obtusifolia don't. So that's a transgressive character because it is outside the range that we see in the progenitors, which just makes me really excited. That's why I love polyploidy, because where is it coming from? We're going to find out. So we have that transgressive delphinidin, which is here. We also have transgressive levels of camphorol in 2 of our species, repanda and Stocktonii, compared to our parents. And if I didn't convince you before, even though these look white, we do have that delphinidin in the tubes of these 2 flowers. And so my questions here are, is this delphinidin coming because of differences in sequence evolution, in differential expression, or a combination of both? And if sequence evolution is important, then is it because we have complementation from our progenitors, where coming together in the same genome means that they can then produce that pigment they couldn't before? Or has there been some sort of resurrection of function during polyploid evolution? Told you you'd see his face again. So we did another transcriptome analysis, again with a early, mid, and late floral buds. This time we had a little bit of better Illumina 2x150 reads that did allow us to do a de novo assembly for our 2 progenitors, which we again combined into our allopolyploid transcriptome. And then Abby, who you've seen before, also mapped these to a specific, just the reads of known flavonoid biosynthetic pathway genes, so we can pull out the sequences of these genes to look at whether they're functional or not. So we found that they tend to not have any nonsense or frameshift mutations. There are no stop codons or frameshifts in any of our genes in Nicotiana sylvestris, the maternal progenitor. We did find that obtusifolia has some either 2 or 3 premature stop codons in ANS, which is our last enzyme in our towards making anthocyanins. But we went and looked at our homolog copies of obtusifolia and our polyploids, and we didn't see those stop codons. We looked at another younger polyploid that also has obtusifolia as a parent, and its obtusifolia homolog also doesn't have these stop codons. So that makes me think that these probably arose in the lineage after it created the polyploids. So it's not really playing a role in the polyploids, but maybe it's playing a role in why obtusifolia cannot make any anthocyanins. So because of a different project, one of my undergrads, who also has graduated, Trinity Tobin, found that the ANS copy of Nicotiana sylvestris retains an intron in its transcripts. So here we have some gels. Here's our genomic DNA from sylvestris. Here's cDNA from another diploid. So we can see that we can definitely get differentiation where we have with an intron, without an intron. But in the cDNA from sylvestris, we tend to have both. So we have some transcripts that have the intron, some that don't. And this also changes over time. So early on in development, we have more transcripts that retain this intron and less that don't. This is not DNA contamination. If we do those same samples in a different gene, we can see that we only have transcripts without the intron in our cDNA samples. So something is going on here. We looked at the sequence. And if you leave that intron in, you get a stop codon pretty much right away. So likely these transcripts with the intron in are non-functional. And so this potentially is playing a role in the lack of anthocyanins in the maternal progenitor. So like, we're going to look into more about this because it's just like, what is happening here? Okay. If we look at our overall expression of our flavonoid biosynthetic pathway genes in our polyploids, we see good correlation. So our transgressive flavanols, we have those two that have higher flavanols, they have higher expression of FLS, which is towards making anthocyanins, and two of these genes that are early on in the pathway, which are going to give more flux through the pathway overall, so they're increasing the amount of pigments that are produced. We also see really nice correlation between increased expression of anthocyanin-based genes, so at the bottom of the pathway, or the transcription factors involved in the two species, the orange one and the cyan one, that produce that transgressive delphinidin. So it looks like we have really good correlation between the transcript differences and our pigment differences in this story, which we can see here, right? So if we look, and apologies for the quality of these graphs, I'm not sure what happened there. If we look at DFR transcripts and correlate it to delphinidin, we have a really nice correlation. Similar to F3'5'H, which brings the pathway to our delphinidin branch, we see that there's an increase in delphinidin with an increase in transcript accumulation. Similar with FLS. If we have an increase in FLS, we have an increase in flavanols. And then we also looked at the ratio between FLS, which brings us towards flavanols, and F3'H, which is going to differentiate towards whether we're making camphorol or quercetin flavanols. And we see a correlation between that FLS to F3'H ratio and our camphorol to quercetin ratio. So we're seeing really good correlation between what we are expressing and what pigments we are producing. So to conclude this story, our ANS gene in both of our diploid progenitors may be non-functional or have reduced function, which may explain why they don't make anthocyanins, but it seems like those mutations happened later after they produced our polyploids. So it's not telling us why our polyploids can produce, or influencing why our polyploids can or cannot produce flavanols. We do have increased expression of early and FLS genes correlating with increased flavanol production, and increased expression of anthocyanin-based genes correlating with the transgressive delphinidin. So it seems like this story is an expression difference story, and less so a sequence evolution story. All right, I have a little bit of time, yes? Just want to highlight a few things that are going on with the students that either have just graduated or are still working in my lab, because I get really excited about the work that my undergrads that work with me do. Lane Jensen graduated a little bit ago. They're actually doing a PhD at the University of Buffalo now. They worked on evolutionary simulations of the biosynthetic pathway. We modified simulations by Wheeler at Smith and changed it so that we could evolve towards having multiple optima to reflect the pigment data we have. And so we can ask questions about what are the mutations that are necessary for flower color shifts to happen, and are some shifts harder to evolve to than others? You've seen Abby's face before. Abby was working on whether positive selection is connected with floral pigment shifts. And so she used GNDS ratio analyses to investigate whether we see differences between anthocyanins and no anthocyanin producing species, and also cyanidin versus delphinidin producing species. So Trinity was working on this other project that then led to us figuring out that we had that intron retained in ANS. She was looking to develop a droplet digital PCR protocol such that we could distinguish between expression of our two homologs in a single reaction. So more bang for your buck. We made some progress. Unfortunately, it didn't really come to pass that it was going to be feasible for what we're doing, but we still learned some really cool stuff along the way. So we've kind of shifted that project with some new folks. I'm going to move this so we can see Alyssa's name. Some new folks who are using ddPCR, but now we're developing two different primers to still get the expression data out, two different reactions this time, but Alyssa Perino and Brooke Tillotson are working on this project, and we're getting into we're taking those 1 million year old polyploids and seeing what's happening with homeolog bias in them. We have another set of folks. Edie Russo has been working with me for about 1.5 years, looking at how cellular dynamics changing over development are affecting the flower sizes that we have in polyploids. And Tim McGrath just joined the lab this semester, so we're excited to see what they're going to come up with. And then finally, I got a grant to go to Kew Gardens in the summer. So full circle, Jeff talked about how that's how my story started. And I got to, ooh, sorry, I get excited. I got to take one of my undergrads to the place where I started my undergrad love of Nicotiana. So it was fantastic. So Marissa McLean and I went and collected a flower morphology photographic data set of a lot of these really old polyploids that have exploded. And we're actually right now, they did it yesterday and this week, and I did it last semester. My intro biolab students are measuring tube length and width from the photographs we brought back, which is super fun. I'm having a great time with it. And then Kendra Moscato joined the lab this semester, and so we're excited to see where we're gonna take that project. So lots of stuff going on. I'd like to acknowledge a ton of people who have made this Nicotiana journey possible, including my PhD and postdoc supervisors, collaborators from all of those places, and especially my undergraduates, because mentoring them is really valuable and really exciting for me, and that's why I do what I do. So I'm excited to see where they go and what they do. Also, some funding sources and computational resources. And with that, thank you for your attention, and I'll happily answer any questions.
Questions for Elizabeth?
How do I?
Do check.
Yeah, I'm trying to figure out how I can make it pop up. Maybe I can get some technical assistance on the Zoom while I answer Rob's question.
Not me, but I'll try. All right.
Thanks, Chelsea. We made it disappear and then put it back. Rob,
First
question.
of all, thank you. That was quite a tour de force.
Sorry, I get excited and talk real fast.
It's a nice lineage for so many reasons. Biogeographically, you have a lot of repeated evolution, the gain and loss of night blooming and color versus white and scent, as you know. And so Sylvestris is a major player in everything you've shown us today. But if I remember correctly, Sylvestris is a problem phylogenetically that's off on a limb on its own, right? And the cladistics are misleading because of the hybrids that it makes. It looks like it's closely related to things that it's actually the progenitor of. So what came before Sylvestris? Thinking about the stop codons and why it's white. Did it have ancestors?
So the stop codons are in Obtusifolia.
Oh, sorry.
It's the intron retained that seems to be my, okay.
But, well, so let's take a step back further than that then. Is the ancestral condition in the Nicotiana to be white and scented, or is it to be colored?
I can't tell you. It's a, I wish I knew the answer.
Because
The way that Nicotiana, or the Nicotiana sections are connected, it means that we have a bifurcation at the base, and that could be white or
that's a
colorful,
coin flip.
pretty much.
Yeah.
Right? And so we do see a lot of shifts in whether things are colored or white in the genus. And so I don't know the answer.
I guess let me follow that with a kind of Stacy Smith question. When is white not white? You know, so many of us thought about white as null mutants, but obviously you were showing us the flavanols can give you white flowers. They're very strongly pigmented and they're antifungal, but they're not nulls.
Yeah.
Right? So if you looked at the tree for the genus, do you think there are many different sources of whiteness?
Ooh. That's a great question. So something I didn't talk about today. And so Sylvestris and many of these do have flavanols, and they absorb UV instead of reflecting UV. Obtusifolia does not have very many flavanols, and it reflects UV. So even within our white flowers, we have phenotypic differences. I haven't managed to do those things with every single, it's my goal to do this with every single species of Nicotiana. Totally not feasible.
But just one more. The Australian ones that you finished with,
yeah,
those have a single origin and they radiate.
Yeah.
So they should all have the same white pigment.
So they, at least in terms of UV absorption and reflection, we were able to do that. I brought my spec with me to London. It was great. And so those all seem to absorb flavanols, or excuse me, absorb UV. We haven't done any pigment data on them yet, but hopefully soon, at least a handful. So in terms of the ones that do absorb UV and produce flavanols, I think those are probably the same white, but then we have some that have a ton of flavanols and some that don't. So like the two polyploids that have that really high levels of flavanols, is that a different phenotype? I don't know. How do we, I don't know. So I guess, again, my answer to your question is, I don't know. Yes.
Yes, thank you. Really interesting. So how did you calculate the age of this? The 6.5 million, it would put you in the Miocene.
So that was not data I did, but they used molecular data and some sort of non-parametric test, if I'm remembering correctly, to estimate times.
Fossils they use to calibrate?
We don't have any within Nicotiana. Off the top of my head, I don't have the specifics, but it's definitely earlier than
Solanaceae?
I think Solanaceae, but I don't have a good. I'm doing great on questions. I don't know is the answer to your question also. But definitely a good point. That's maybe we don't have specifics, like maybe our exact dates aren't great, but I think our relative dates are certainly good. So even if we don't have the exact dates, we still have that trajectory of understanding which ones are younger and which ones are older. So that still kind of fits with my, I wish we had more fossils, is again the answer. I saw a hand over here and then you're on deck.
It's a naive question. A lot of these pigments that you've talked about appear in vegetative organs as well as floral organs. Is it a fair supposition that what you found in the petals is also happening in the vegetative organs?
I think yes and no. For the flavanols, I would assume yes. I think it's way more likely that those vegetative tissues have flavanols compared to the anthocyanins. Just by looking at the vegetative tissue in some of these plants as I've worked with them, there are some that I call them, they have purple leaves. People are like, no, that's green. I'm like, yes, but it's purpler than these ones. So I think that maybe some of them have some anthocyanins in them. But I would certainly guess flavanols. The levels and the ratios of those things, I'm not sure. But even, hold the phone, even within the flower tissue, can I find that? Yeah. So even in the flower tissue, we have differences. So the pigments are really concentrated in the limb and not in the tube. So I do think that for the anthocyanins, at least, we have really specific expression of when we're turning those pigments on, which is probably not happening in the vegetative tissue. That answer your question? I got one. Okay, yes.
No, you see subgenome dominance in the older, like natural polyploids. Is subgenome dominant?
We see a more expression bias.
Okay.
So at the gene by gene basis, we see a greater deviation from equal expression. But if we look at it overall, we don't actually see subgenome dominance.
Okay. Is that a function of the age of the polyploid? If you could look at that gradient of the ages of those polyploid events, you see with older polyploid events more of that bias? That's
a great question. And it's part of I'm trying to piece this together, right? I'm working in all these different aged polyploids, and hopefully at some point I'm going to be able to put all of that together.
And I guess to follow up to that, is the logical conclusion of that subgenome dominance and maybe a contraction that you see?
Potentially. Yeah, definitely. At least in, we don't have full genomes of all of these polyploids, but for the sequences that people have used phylogenetically, we do seem to have both copies of things in most of these polyploids. Maybe the oldest ones were starting to lose some things, so maybe that's when fractionation is starting to happen, but we don't have a good answer to that yet either. But definitely a great question, and kind of fits with this idea of how can we use this trajectory to really answer cool questions. Yeah.
What about flower size and shape?
What about it? Tell me. Yeah, I also care about those things.
There's such a difference that it's be playing out in interesting ways, I guess I would say.
Yeah, and we're trying to get at that in some of the things that that Edie and Tim are working on with the cell stuff. Also, the Suavilantes project, we have more differences in shape, and size than color in those. So those are things that we're getting into. That again, I don't have good answers to your questions, but I would love to find out. I guess I can tell you a little bit, right? We do see that we tend to have wider tubes in really young polyploids, and that seems to be because the width of the cells are bigger with polyploidy. So polyploidy increases the width of the cells and increases the tube, which might influence pollination. So maybe polyploidy potentially is giving us more generalized pollination. But what Edie and Tim are looking at is the cell size in the older polyploids. And preliminary data, it looks like we lose that in our older ones. So we're reducing the size in our older polyploids, potentially like we're reducing genome size. I'm excited to find out more about that. Jeff and then Rob.
What's the link with pollinators in the sizes and the pigments? The amount of flavonoids for reflection for bees seeing bee versus bats not seeing.
Yeah,
What's
we
the correlation?
We have some pollinator data. We have pollinator data more for diploids than we do for polyploids. So that's another whole trajectory that I would love to take my research program in the future, is to look and see how these morphological and phenotypic differences affect pollinators. But unfortunately, we don't know a whole lot about what's pollinating our polyploids. So again, I don't know the answer to that question, but that's great. I have so much to work on. Rob.
Coming back to Adrian's question, you also have a symmetry issue, right? Like tomatosifolia and Otophora are zygomorphic. We know what genes are responsible in a lot of plant families for zygomorphy. Are they segregating between the hybrids? Because Sylvestris does not look zygomorphic to me.
No, Sylvestris does not. I would say that most Nicotiana are not. There are a handful. A lot of the alate are also zygomorphic. Trying to just think about all the polyploids in my head for a second. I think most of them are radial, at least in the limb. There are some in the older polyploids that we were just investigating where they do start to have those. So again, really great questions that we can investigate. We know those genes. Hopefully with more data, we can, again, start answering those questions. Really great ones.
And that gets to Jeff's question too, because certain kinds of pollinators like flowers that have an up and down.
Yeah, definitely. Yeah, go for
The
it.
cell size question. It could be cool with more data to see if there's a relationship between polyploidy and endoploidy. Maybe you can answer that. If in these polyploids that maybe you see less, or if it's compensating, or they're going to get their genomes one way or another. Right?
Yeah, I don't know. Again, really cool things that hopefully be able to investigate at some point. But yeah, there's so many cool things. Other questions, folks? Yeah, go for it.
Because you made your own de novo transcriptomes, were you able to see any bias when you took that same data? And I'm assuming you did a concatenated transcriptome in the mapping. So were there any reads that maps to one or the other incorrectly that you saw?
Yeah, and that's part of why I didn't tell you a story about homolog bias in Repande, because we have, I need two hands for this. I need two hands for everything. We have a threshold for when we're looking at those homolog bias where we can, have to have a less than 10% mismatch between our two copies. And for many genes, we reached that. For some, we didn't. Luckily for the tobacco story, we reached that for all of our flavonoid biosynthetic pathway genes. We didn't hit that for Repande, which is unfortunate. I'm hoping maybe Jacob has a better answer to this question. Like if we add more long reads to those progenitors, we didn't have that for this one, whether we might be able to pull out the differences. We also have the added difficulty of there's 4 million years of evolution in the polyploids and in the diploid lineages in that system. And so being able to really figure out homologs, because the homologs within the polyploids have diverged in sequence. It's from the progenitors. We see that way less in tobacco. It happens sometimes, but they're pretty tight, so that's way easier. So yes, and then we just can't ask those kinds of questions for those genes. Does that answer your question?
Yeah.
Awesome. Other questions, folks?
I do one real quick. So that,
One real quick.
that gets into like the next, I keep thinking about is in the long-term one, right? We've got this 90 species or whatever it is, thinking about copy number, right? And whether you get retention of homologs over time and how that might lead to either sub or neo-functionalization of those copies.
Yeah.
Can you see how that you might attack that across those different timescales?
Yeah, I think it'll probably have to be on a like candidate gene basis, more likely. Maybe not all the things, but at least, like I said before, folks have used some genes to look at phylogenetics, and we sometimes don't see the second copy in some of those Swabulantes, the old ones. It's certainly interesting. I mean, we're already seeing some pseudogenation in young polyploids sometimes. And I didn't talk about it today, but when we looked at all of those flavonoid biosynthetic pathway genes in the Rapandae, there are a few more. Like some of the homologs within the polyploids have frame shifts. And so we're starting to see some of those things. But yes, again, all of you have really great ideas. These are really good questions that with more data will definitely be interesting to investigate.
Right, well, let's thank Elizabeth.
All right, thanks, folks.
This has been a production of Cornell University, on the web at cornell.edu.